This ability for us to make energy rests strangely on this 5 micrometer oval organelle known as the mitochondria. What are mitochondria?
Mitochondria are the power plants inside of all of our cells. And with a decline in mitochondria, you have a decline in functionality at the organ level and also on the whole human body.
Welcome to Commune. We are on a mission to inspire, heal, and bring the world closer together.
Okay, Chris Rinsch, welcome to the Commune podcast. Great to be with you, man.
Hey, great to be with you, Jeff. Thanks for having me.
Yeah, well, we had the opportunity to meet in person at a party in Commune, Topanga when our pomegranate trees were in full splendor. I remember in late October.
It was amazing. Yeah.
And if my memory serves me correctly, I believe we were the last one standing at that party, which probably speaks to our mutual bioenergetics, if you will.
Exactly. Our mitochondria were working full speed.
Yes, they were. Yes, probably with some extra enhancement. Yeah, I'm really looking forward to today's conversation. I've been thinking about it. And really, just the state of being alive is contingent on our ability to produce energy. In fact, on some kind of philosophical level, we're just kind of animated information, kind of links in this continuous chain of energy transfer. But this ability for us to make energy rests strangely on this 5 micrometer oval organelle known as the mitochondria. And it's so cool that the mitochondria has really become more and more part of the zeitgeist, if you will. And you guys are on the tip of the spear in terms of creating more fluency around this strange little purple bacteria that got engulfed by an archaea some 2 billion years ago. And maybe we'll talk a little bit about the history of this organelle. But I'm excited to jump into all the roles of the mitochondria with you, its relationship to aging, and of course how we can upregulate the functionality of our mitochondria, specifically as it pertains to muscle function, but also as it pertains to brain health, and everything that you guys are doing at Timeline and with Mitopure. So excited to jump in. So maybe at the very, very, very basic top level, what are mitochondria?
Mitochondria are basically the power plants inside of all of our cells. And so essentially what they're doing is producing the energy, the ATP that is needed for all of our cells to function normally, for all of the different biochemical reactions to take place inside of the cell. And so they're essentially this battery that we need to make our cells function and work properly. Without functioning mitochondria and with a decline in mitochondria number or functionality, basically there's less energy inside of the cells, so less of the cell's functions can take place, and so that sort of transmits itself not only through a cellular level, but on sort of a whole tissue level and on an organ level. And so you have a decline in functionality at the organ level and also on the whole human body. So it's very important for for us to function properly, and we need that energy.
Yeah, and so just break it down from almost a very basic level. Mitochondria exist inside the human cell. Is that correct? And is there a certain prevalence of mitochondria in certain cells versus other cells? And how many mitochondria are in a cell?
Sure. Well, yes, mitochondria are located inside of the cytoplasm of the cell, and certain tissues require more energy than others, and so there tends to be more mitochondria and a higher density of mitochondria. And so particularly skeletal muscle, which uses a lot of energy, as well as cardiac muscle and even the brain. There's quite a high density of mitochondria.
And are we talking 200 mitochondria per cell, 2,000 mitochondria per cell, or am I not even in the right ballpark?
Well, I think it really depends on the tissue, but it's not in the tens of mitochondria. It's more in the hundreds and the thousands of mitochondria, and that just depends on the size of the cell too. So, yeah, they're very small, so you can pack a lot in.
Yeah, and they tend to cluster, right? So, I remember from biology class, generally you'd see this mitochondria as kind of this jelly bean like image inside the cytoplasm of the cell. But in reality, they tend to cluster in groups. Is that right?
Yeah, they do. You do tend to find them sort of stuck together. We've looked at transmission electron microscopic images in some tissues, and you can see lots of mitochondria kind of sticking together, and you can also see the structure of those mitochondria, the cristae inside, and how that cristae formation changes with time and during aging as well.
Mm-hmm. And, the mitochondria in some ways are where food and oxygen meet on some level. So people are somewhat familiar with metabolism and digestion and how macronutrients get absorbed into the bloodstream and then brought to the cell. Glucose, for example, gets ushered to the cell by insulin and then goes through glycolysis and then eventually into the mitochondria for the last couple stages of cellular respiration. We can maybe get into that. And then, oxygen is also obviously comes through the lungs and brought through the body through hemoglobin in the blood and then transferred out also into the cells. And it seems like the marriage of glucose and nutrients that we're eating for energy production and the oxygen that we're respirating meet and have a tryst at some level at the mitochondria. It's pretty fascinating.
Yeah, it is. I mean, you have you're breathing in your oxygen, and it's basically for the mitochondria, and then you're eating all these foods, and the fatty acids are then transformed through the mitochondria into ATP through the electron transport chain. Yeah.
Yeah, and so mitochondria, the catchphrase that most people use is, they're the powerhouse or the power plant of the cell. But mitochondria are also involved in a lot of other functions as well. Do you mind touching on some of the other roles that the mitochondria play?
Sure. So mitochondria are also involved in various signaling pathways inside of the cell. And so mitochondria can trigger the cell down a pathway of cell death as well if the mitochondria are significantly damaged. And so it's an important aspect that we don't think about, of all of the different roles of the mitochondria in the cell, although providing the energy and having proper functioning mitochondria is essentially key to have all of these processes functioning normally.
Yeah, we would not be moving our lips talking to each other right now if we weren't unlocking the chemical energy in food through this incredible process, and then being able to transfer that energy into mechanical or kinetic energy, or et cetera. So, you brought up something interesting around cell death or apoptosis, or as it specifically pertains to mitochondria. When mitochondria get damaged, there is this process called mitophagy. But perhaps we can explore why mitochondria start to become dysfunctional in the first place. So as I understand it, part of cellular respiration, of essentially the creation of ATP, a byproduct of that is the production of some free radicals or reactive oxygen species that can at some point damage the cell and the mitochondria. Can you pull on that thread a little bit?
Yeah, sure. So, throughout the electron transport chain, as you're converting your fatty acids into ATP, essentially you generate a lot of reactive oxygen species, and in the process, these reactive oxygen species they will act at the level of the cell membrane or the mitochondrial membrane to damage that, and as you're causing the damage to the mitochondria during the process of making ATP, essentially inside of the cells, you have a process by which you are taking the mitochondria and you're repairing the mitochondria. And you alluded to it earlier, this idea of mitophagy. It's mitophagy, sort of the cell eating the damaged mitochondria. And so essentially what happens is that the damaged part of the mitochondria is butted off from the healthy part of the mitochondria, or if it's an entire mitochondria, it will be targeted on this pathway in which there will be it will be engulfed by and then digested into its component parts. And so these component parts can then be essentially recycled inside of the cell and go into creating new mitochondria and growing our existing healthy mitochondria. And so it's a cycle that's ongoing, and it's not just sort of happening at one time, but it's a continuous process. And the cells need this in order to stay bio, say functional from a bioenergetic perspective.
Yeah, it's fascinating. I mean, we often hear a lot about autophagy, and that can be associated with fasting and the triggering of certain cellular pathways. And autophagy is a more general term, technically meaning self-eating, but really referring to kind of the breakdown of dysfunctional cells into their amino acid component parts. And then what's amazing is that then the body can reuse or recycle those component amino acids for the rebuilding of new proteins, which is just always boggles my mind, the kind of foundational intelligence of the body there. But that's also taking place at the mitochondrial level with mitophagy. Is that right?
Yes, that's right. So mitophagy is really a sort of a subset almost of autophagy. As you were mentioning, autophagy is the term to describe the general recycling of proteins, of aggregated proteins or damaged organelles inside of the cell. And it is really quite miraculous that inside of cells you have this process that is basically a cleaning process that enables the cells and all the cells' machinery to stay functional, so that the cells just don't die after a certain amount of time. And the particular process that we're talking about, mitophagy, is really essential for the mitochondria, because the mitochondria are getting constantly damaged as they produce ATP. And so if we can, and one of the problems, and we'll get to this, is that as we get older, this process of mitophagy is not can decline basically, and can be disturbed not only due to age but also due to our general lifestyle choice, our activity, our diets, and so it's all of that is very important to maintaining the proper bioenergetic function of our cells.
Yeah, I definitely want to excavate all of the detrimental behaviors that we engage in to downgrade our mitochondrial function and just our ability to produce energy in general. I do want to poke at a couple other mitochondrial roles, because as we touched on, the mitochondria does produce a certain amount of reactive oxygen species. These highly reactive molecules that can cause damage. But at the same time, at the mitochondrial level, there is the production of certain antioxidants to balance, in the best of all worlds, the creation of these reactive oxygen species. So, can you talk a little bit about like glutathione and even melatonin at the mitochondrial level and how they work to mitigate the production of free radicals?
Well, yeah, I mean, you have, as you mentioned, glutathione is very important as an antioxidant. It's a very powerful antioxidant in the cell, and that's one of many antioxidants that are important to keep the balance of the free radicals and in check, so that you have your mitochondria, let's say, stay healthy despite all of the stress of creating the ATP along the way. But they're not enough. And I think that's the message, that just by, let's say, supplementing and providing more antioxidants, you can't really stall the inevitable, which is the damaging of the mitochondria, just as a normal process in the day to day as the cells are functioning.
Yeah. So, just because the history of the mitochondria is so fascinating and interesting, I wonder if you could just spend a couple minutes touching upon how humans and I suppose animal life in general came to co-evolve with these energy-producing organelles.
Okay, I think that's an area where I'd prefer to pass that off to an expert.
Yeah, fair enough. Fair enough. Well, we can even just skirt over it. I was reading something fascinating that Lynn Margulies, who was Carl Sagan's wife, and then wrote under the name Lynn Sagan, was the first one to posit the notion that mitochondria actually came from this purple bacteria, I think sometime in the 60s. And she wrote a paper on it, and no one would publish it. And finally she got it published somewhere after being rejected 7 or 8 times. And it postulated this notion that, hey, the mitochondria looks so much like bacteria under a microscope. And if you just go on images, on Google Images, you can anyone can do it. You can just see, and there's an incredible resemblance, between the two. And then, there started to be these other characteristics that they share, because the mitochondria actually has some of its own genome. Is that right?
Sure, and mitochondria does have its own genome, although there are proteins that are coming that have that genes also from the cell. So they sort of live together, and over time, as you're mentioning, that the mitochondria and our cells, there was that evolution where the mitochondria basically, from the bacteria, became included inside of our cells and played that role of generating the ATP and the energy for our cells, and also taking in the oxygen. So this coevolution, yeah.
Yeah, I think it's fascinating. I think about the microbiome, for example, which is such a hot topic, and of course, once we get into our conversations around urolithin A, we will probably circle back to the microbiome, because gut bacteria plays a significant role in the kind of metabolism of certain polyphenols to create endogenous urolithin A. But before we get there, I think about the human, what it is to be human, and we really co-evolved with all of this bacteria in our gut. So we're starting to know about that. We've got 39 trillion cells, mostly in our gut, but kind of now we're learning kind of all over our body, particularly like in our nose and in our mouth, even on our skin, known as the microbiome. So we co-evolved with these prokaryotes over here, and then we start to learn about the mitochondria, which seems to be a bacteria that an archaea tried to eat, but the bacteria resisted the digestion of it, and they found some sort of symbiotic relationship. And over time, as the world became more aerobic and there was more oxygen in our atmosphere, it became more viable for these particular single-celled organisms to exist and to create energy, and somehow they became absolutely critical to the expansion of complex life, because we just didn't have the ability to make enough energy, before the advent of mitochondria. We were simply kind of these single-cell organisms, or I guess photosynthesis had started to happen, but really it's this incredible endosymbiosis that then gave birth to all of this amazing life that we see in the world. So it's amazing. So we know now that mitochondria play key functions. What is mitochondrial dysfunction, and what are some of the components that contribute to that dysfunction?
Yeah. So when we're talking about mitochondrial dysfunction, it's really a question of back to energy and the ATP generation, and we've looked at this clinically, where we with a tool called magnetic resonance spectroscopy, and what it allows you to do is look at energy production, in fact ATP levels. And so you put it in a special magnet, you contract your muscle and you can see the decline in the energy, and then when you stop, you can see the return to normal. And depending upon the health of the mitochondria, this is quicker or it's slower. And so, to summarize, depending upon your condition, and even your age, you could have a decline in mitochondrial function, and that can have an impact directly on limb function, limb muscle function, for example, mobility, or other type of function. In fact, there's a number of diseases that are linked to a decline in mitochondrial function, and one of those is sort of skeletal muscle component, and there's others that are even at the level of the brain and brain health as you get older. And so yeah, the decline in mitochondrial function can have a number of negative effects on our lives.
Yeah, I recently spoke to a doctor named Chris Palmer out of Harvard, who wrote a book called Brain Energy, and fascinating book. And his unified thesis is really that mental disorders are really metabolic disorders at the mitochondrial level. And so essentially, I mean, and it makes some sense. I mean, if you begin to suffer from some form of neurodegenerative disease, I mean, what you're seeing is brain shrinkage and the neurons' inability to properly create energy. And so, his thesis is fascinating. But, as far as a lot of your work is concerned, I think you guys have focused largely on muscle function. And can you talk a little bit about sarcopenia and some of the detrimental impacts of loss of muscle function?
Sure. We did very early on. We conducted a study looking at a population that are considered pre-frail, and so that's not yet at sarcopenic levels, but it's basically individuals who are starting to have problems stepping, walking, getting out of chairs, and general mobility problems, and we compared those with a group of age-matched individuals, and this is age of around 70 years old, who were athletic and exercising on a regular basis. And we tried to understand, well, fundamentally, what is different between in the muscles of these two populations. And so we went and we ran the study that I was referring to earlier with this magnetic resonance spectroscopy, and we saw a decline in the mitochondrial function in the legs of these individuals who were pre-frail. And then we did biopsies, and we looked at the biopsies, and we saw a decline in the gene expression of mitochondria genes, and showing that there's basically less mitochondrial biogenesis, which that means production of mitochondria, sort of a regular production of mitochondria. And we saw a decline in the various complexes, and this is the mitochondrial complexes, which are important in the production of ATP. And so that was one of our first observations that it's that mitochondria, the decline in mitochondrial function, is really essential for healthy muscle function and mobility. And now this wasn't sort of a cause and effect study, but it's an observation in this population that were significantly impaired as being pre-frail, that mitochondria, when looking at comparing them with healthy, that mitochondria gene set was one of those leading gene sets that jumped out as being impaired. And when you think of now, we haven't looked at sarcopenia. Sarcopenia is taking it another step, where you're looking at the amount of muscle mass and how that declines with time. And so sarcopenia is where there's been more muscle mass loss. So it's not only a loss of function, but also a loss of mass. But I think that, and there's always been historically an approach when you have a decline in muscle mass that you try and increase the muscle mass, which makes a lot of sense. But then you need to make sure that increased muscle mass is also very functional. And so, and it's not easy to increase muscle mass, certainly as you get older. And that's one of the challenges. Although I have seen some people recently who are in their late 70s and who have lots of muscle, and it just is super impressive. I don't know how they do that. But yeah, when you're when you're having whatever amount of muscle you have, you want to make sure that it's basically functioning at its peak. And I think that's really where the mitochondria comes in. If you can take your muscle cells, even in a sarcopenic individual who has had a decline in their muscle mass, and make them function better, then the muscle at itself should function better. And so that's one of our underlying approaches to improving muscle function. It's by acting at that level of the mitochondria and basically optimizing the bioenergetics of your cells. No matter what the sort of age you are, or what the statuses of your tissue are, whether they're, you have large muscles, whether you have small muscles, to try and sort of raise the functionality across everything.
Yeah. Um, yeah, it's interesting. I mean, you guys are doing like muscle biopsy. Are there simpler, kind of more subjective measures of poor mitochondrial health that one, might just discern on their own? For example, is chronic fatigue or brain fog, a reflection of mitochondrial dysfunction, or I don't know, like grip strength or
Well,
your
well,
walking gait or whatever.
yeah. So there's a lot of so mitochondrial function is really linked to physiological performance. So you can screen people for performance using different types of measurements, and we spoke about walking speed. There's the there's tests that allow you to screen for people who are let's say pre-frail or more frail by just the 6-minute walk is a common one. How far can people walk in 6 minutes? There's also different measurements, like getting them on a bicycle, for example, and seeing what what their performance is on a bicycle. And this is more non-invasive way, of course, to screen than biopsies. And there is the overall energy level too. There's we think, we're our general energy level is linked to fundamentally the energy level of our cells in our body, right? And so if our cells aren't, and the mitochondria aren't functioning well, you can't expect your energy level to be very high. So, and you were also speaking earlier about brain function. There's been, in addition to memory and these types of cognitive effects, there's been links to stress and the importance of properly functioning mitochondria for to help resist stress that you might have. So yeah, there's a number of different ways that you might feel a change in your mitochondrial function, even when you're exercising. How easy is it for you to recover after post-exercise?
Yeah. So you mentioned stress as a contributing factor to mitochondrial dysfunction. What are some of the other prevalent contributing factors that cause your mitochondria to dysfunction?
Yeah. So in addition to stress, I would say that aging is one of the primary drivers of mitochondrial dysfunction, just generally speaking. And one of the reasons is that as you get older, this process that we were talking about before, called mitophagy, actually declines. And so the cleanup of your damaged mitochondria is slower, and consequently you have more damaged mitochondria accumulating inside of your cells. And so generally speaking, on a whole, your cells aren't functioning at their peak. And but then there's also lifestyle choices. It's known that when you exercise, that this is stimulating a process of mitophagy as well. And so you'll see a lot of you'll see mitochondrial biogenesis if you take biopsies in people who are exercising regularly. You'll see an elevated level of mitochondrial biogenesis and this creation of new functioning mitochondria, versus those people who have a more sedentary lifestyle, and and you'll see less of that going on. And so in general, if you were to look at those having a sedentary lifestyle versus a very active and fit lifestyle, the mitochondria are going to be much more active in those and much more functioning in those people who are active.
Yeah, so exercise versus sedentariness plays a big role there. What about sleep?
That's a good question. I'm not really familiar with the impact of sleep on mitochondrial function, but I would say that there's one thing that we have to think about when we're sleeping, that there is this whole aspect of dietary restriction and fasting, and fasting is another element that is stimulating mitophagy and keeping our mitochondria healthy. And so this and you can, when you look at the profiles, the genetic profiles in terms of on the muscle cells and on other cells when they're fasted, you see an upregulation of mitochondrial biogenesis as well, and an improvement in mitochondrial function. So it would be logical to think that if you're sleeping for a longer period of time and getting more restful sleep, that you're probably favoring a better mitochondrial profile inside of your cells.
Yeah, I mean, I leverage the 16-8 intermittent fasting protocol most of the time. Although we just finished a very busy holiday season, I can't say that I was fundamentally strict over that period. But yeah, if you use, your sleep window to chalk up, let's say, 8 hours of fasting, it certainly makes, the other 8 hours or however many hours you end up fasting, a lot easier. And so, there is a component here of fasting stimulating certain cellular pathways, like AMPK is one, for example, that then triggers kind of this cell repair process, autophagy and mitophagy. It also seems like sleep has some relationship to insulin sensitivity, and poor sleep seems to contribute to insulin resistance. And so I just kind of want to touch on that for a second, as it pertains to diet and lifestyle. So, obviously there's this kind of standard American diet, which is very high in refined sugars and refined grains and starches and ultra-processed foods, that is contributing to high blood glucose levels and then kind of down the line, insulin resistance, which has an impact on our mitochondria's ability to create energy. Is that right?
Yes, I mean, it's these diets, as you were referring to, are really not optimal for your not only for your body but for your general metabolism for your mitochondria function. And it's not only the what we would assume is logical that these types of diets aren't good for you, but there are foods that you can take that are beneficial for your mitochondria, and I think that's where we should be thinking of how can we change our diet or incorporate things into our diet that are going to be helpful to help maintain that an optimal functioning of your mitochondria, and eventually stimulate this process of mitophagy.
Yeah, exactly. So this is the good news. This is the gospel.
Yeah.
We've covered a lot of the bad news, and all of the different components to dysfunction. But the good news is that our body has adaptive mechanisms that, under the right circumstances, can stimulate processes like mitophagy, or what you referred to, mitobiogenesis, the creation of actual new mitochondria in the cell, which is good news. And just as more kind of generally as it pertains to metabolism, you can increase your insulin sensitivity. You can, reverse insulin resistance and become more metabolically flexible. So there's a tons of different things that one can do. And some of that you already mentioned. So there was, exercise and fasting, but maybe you want to touch on, some of the foods and their constituents that can help trigger some of these healthful processes.
Sure, Jeff. So what we've been looking at, we started looking at the pomegranate quite a number of years ago, and one of the types of compounds, it's actually a class of compounds that's found inside of the pomegranate, are the elagitannins, and these are larger compounds, phytochemicals that are found not only in the pomegranate, but they're found in various nuts and berries. And essentially what happens is that when you consume them, that they're digested in the stomach, and then they're processed in the intestine by gut bacteria. We were talking about the importance of microflora earlier. And if you have those right gut bacteria, these compounds, these elagitannins, are processed into a postbiotic that is called urolithin A. And we've spent a lot of time now studying urolithin A, and together with a number of collaborators at Scientific Institute here, the EPFL, as well as other institutes. And what we've shown is that this postbiotic, urolithin A, it can stimulate this process called mitophagy. And so this is pretty exciting because this is something that's coming from the foods we eat, and it has this potential to interact at the cellular level and basically help reduce and reverse the decline of mitochondrial function that happens naturally, and by boosting this level of recycling the damaged mitochondria. And so we've spent a lot of time studying this, and so this is something I think that it'd be very pertinent for people to take. It's the first, and to my knowledge, the only nutrient that is actually stimulating this process of mitophagy when consumed orally.
It's fascinating, and I'm so curious to know how you landed on pomegranates. But I just want to make sure that I understand the mechanism of action here. So, if you consume exogenously, like pomegranates, and we can talk maybe about the part of the pomegranate that has the elagitannin compounds, but also maybe raspberries. I think walnuts and pecans maybe have elagitannins as well. And they go through the normal process of digestion, but some of that goes into essentially your colon, your large intestine, and gets metabolized by certain strains of bacteria, and postbiotics or metabolites are produced by that bacteria to produce this compound called urolithin A that seems to have a really, really positive effect on mitochondria and, as you say, mitophagy. Is that a general understanding of what's
Yes,
happening?
it's quite as you say, it's quite a pathway. It's quite a process from the foods that you take to
Right.
actually getting this compound.
Yeah, that says nothing of actually getting into your car and driving to the farmer's market to get the walnuts in the first place,
Exactly.
or the pomegranates,
Exactly.
or to grow them. So, is there a particular bacterial strain or species in the gut that synthesizes urolithin A, or do we even know what that might be, or is it?
Yeah, it's very interesting. We've looked at this over the years, and it's very complicated to basically isolate the right strains that can perform this conversion. And there's been a couple of groups out there that have postulated that some strains that could be acting on it, but it's been yet to sort of single out an individual strain that will perform this activity, and it's probably due to a combination of bacterial strains that are playing different roles in terms of the chemical modifications, because it's not simply it's creating something, but it's have a starting compound that's coming in and it's being chemically modified, and you're having different modifications, and so this is makes it a little bit more tricky.
Yeah, I've got to retrain my reductionist Newtonian brain. I always want to know, like, what is the specific, bacterial species that, metabolizes alagitannins into urolithin A. You're absolutely right. It rarely works that way. It's just, these bacteria are working within a mosaic and a matrix, and it's probably very difficult to put your thumb exactly on the process. I mean, I assume, though, in general, people have to have a very healthy gut microflora to be able to create, this postbiotic, and probably not everybody has the ability to synthesize urolithin A at the gut level.
Yeah, you're right, Jeff, that we actually conducted a clinical study a couple of years ago in Chicago, where we had people take pomegranate juice and consume a glass of pomegranate juice, and we monitored their ability to basically produce urolithin A, and we took blood samples, and we measured urolithin A in those samples, and essentially what we saw was that only about 30 to 40% of the population was able to produce measurable amounts of urolithin A, and so they were measurable, but they weren't, based on other work we've done, they weren't at the level that you would need to actually have the benefits that we see with urolithin A when it's administered in a more direct manner. So
that's interesting. Yeah,
and then
yeah.
Sorry to interrupt, but I think one of the challenges also with taking pomegranate juice is just the high amount of sugar. And during that study, we compared the amount of urolithin A that you get into the body with a glass of pomegranate juice versus directly administering 500 milligrams of urolithin A. And what we saw was that you needed basically 6 times more pomegranate juice. So you basically need to have about 6 glasses of juice to get the same levels as directly administering. So it's not an effective way to get what you need to give you those benefits.
Yeah, it sounds like it might make you pre-diabetic in the meantime too. Exactly. I wear a CGM, and I can only imagine what 6 glasses of pomegranate juice would do to my Levels app. But so I think that's fascinating. So really, only 30% or 40% of the people can even synthesize urolithin A from the alagitannin, even in the best case scenario, and potentially that points to people's compromised gut flora, but it's also maybe just bio-individualities. Some people just don't have the ability to generate certain compounds. Also, I suppose when you're drinking the pomegranate juice, you're really, through the process of processing a pomegranate, you're really you're losing a lot of the white pulpy part of the pomegranate. And is that where you find most of the alagitannin? Or?
Yeah, so that's back to your point of where are the origins sort of located inside of the pomegranate. I mean, you have your arils that are bright red, and then you have this sort of yellow whitish membrane that that sort of surround and all of those, and and it's really this yellow whitish membrane that's very bitter when you taste it, that contains all of these elagitannins. I don't encourage you to bite in and chew some of that because it's not enjoyable at all. But in the process of basically making pomegranate juice and compressing the pomegranate, you squeeze out these elagitannins from this yellow membrane, and that goes into the juice, and so then you are able to consume it. And so we've actually measured levels of elagitannins and juices and some commercial juices, and they do seem to be pretty consistent. So there is a very consistent processing of the juice, although it's it doesn't give you enough, and you still have that whole process that we discussed of converting the elagitannins into urolithin A, and then having that actually get absorbed into the body.
So you've been able to synthesize a kind of pure urolithin A
Yes.
compound, and I'm curious, how do you actually do that? I don't assume that you have a bunch of petri dishes with bacteria in them and feeding them elagitannins. So is there a process in the lab by which you create pure urolithin A?
Yeah, so it's a very clean process. It's industrially sort of similar to the way like a vitamin C would be manufactured. So this is using synthetic chemistry where we make a product that is ultra pure. So likely much purer than you would be able to purify out if you were to try and extract urolithin A from some type of a fluid, for example, a body fluid, after you would produce that normally. So yeah, that's and the nice thing is that you have that you can then titer out into very specific doses, and then you can analyze the impact of specific doses on mitochondria orally. And so that's what we've been doing.
And what is the proper dosage that you would recommend for the Mitopure product?
Yeah, so the product that we're selling is Mitopure, which is our proprietary urolithin A, and the dose is 500 milligrams, and that's sort of the starting dose. We've done a couple of studies now. Actually, we've done 3 double-blind placebo-controlled studies with Mitopure, and what we've shown is that at 500 milligrams, we can see that benefit on the mitochondria function, and that we also see a benefit on muscle functionality. And so we've looked at, we've had our very first study, we looked at 500 milligrams and a gram after a month, and what we saw was an improvement, and this is through biomarkers. We were speaking about muscle biopsies. We looked at muscle biopsies before and after taking our product for a month, and what we saw was an increase in gene expression of mitochondrial genes, and sort of a gene set that's an enriched process. So it's what they call a mitochondria gene set. So it's a number of genes that's involved in the normal mitochondrial health and functioning, and you see all of these genes upregulated, and together they have the combined effect of improving and increasing the mitochondria gene set and enriching that inside of the cell. And so that was one of our very first studies. And then the second study we conducted was in individuals who were 40 to 65 years old and who were sedentary and had a little bit more overweight. And so we looked after 4 months. This is the first time that we actually investigated the effect of urolithin A on a physiological endpoint. And so we looked at the muscle function after 4 months, and we saw an improvement in leg muscle strength by over 10% in those people who were taking 500 milligrams and 1,000 milligrams, and at the higher dose, we were beginning to see some effects on VO2 peak and and even on 6-minute walk. And we continue to run these other studies where these other biomarkers, as I mentioned, the biopsies, and also looking at basically blood biomarkers that you'd find in the plasma. And so what we've consistently seen is that when we look at biopsies, we see an improvement in mitochondria function through this gene set enrichment of mitochondrial genes. And in terms of biomarkers, we also see an impact on biomarkers, particularly acylcarnitin. And acylcarnitin basically is downregulated, and that's what we see sort of when we compare the before and after snapshots in individuals who've been taking urolithin A. And so this has been shown in the past that people who have more of an impaired mitochondria function, that they have elevated acylcarnitin levels, because this is part of the whole metabolism process of the cell in terms of consuming those acylcarnitins.
Fascinating. And at this juncture, do you understand the mechanism of action of urolithin A at the mitochondrial level? I mean, certainly you guys have been unbelievable in terms of your commitment to clinical research and to, creating a product that's been validated by science. But I wonder if you actually understand what the mechanism of action is at the mitochondrial level. I mean, what is this urolithin A compound actually doing? Do we know?
Well, it's a good question, Jeff, that we are hard at work on, and I think your question alludes to sort of what are the molecular interactions of urolithin A inside of the cell, and what are the cascade of events that occur that leads to mitophagy. And what we can say is one of the ways we look at mitophagy and sort of determine that there's mitophagy happening inside of the cell is one of many, but it's looking at ubiquitinization of your mitochondria. And so basically what happens is that your mitochondria get, when they're damaged and they're targeted for this process of mitophagy, they're become sort of decorated with ubiquitin, and this targets the mitochondria to that pathway. And with regards to all of the other sort of processes that are involved, from the urolithin A arriving to the cell to that, we are still working hard at that. There's, in fact, this whole process of mitophagy. This is relatively new over the last 10 years that people have been working on mitophagy, and it's a pretty new field with a lot of really smart academic groups working on it and trying to understand what are the different ways of mitophagy. And I think one of the reasons that we think that we've been rather successful in our stimulation of mitophagy is that it's a gentle stimulation, because there are other pathways that are stronger. And the fact that we're dealing with urolithin A, which is basically this nutrient that's been that we've been exposed to since the dawn of time, This is very, very safe, and there's no there's no risks associated with it, which make it so that it is having this gentle but yet potent effect on our cells, and yeah, we will see more in the future as we continue our research along that way.
Yeah, I mean, you mentioned that the risk profile is very low, no side effects, but you guys have also been stringent in terms of going through all the processes and the GRAS process and all of that. So you guys, and also I'll just mention, I think you guys have been around as a company since 2007 doing clinical research. So this is hardly a product that's just gotten thrown on to the shelves on a whim. This is a product of 15 years of research, which is admirable.
Yeah, well, thank you. I mean, I think, and as you say, it's something that we've worked on a preclinical and a clinical manner for over these last 15 years, and this is, I think, what we've taken is more of a traditional a biotech mentality of let's raise the bar in terms of the level of validation that you need, and the science, and because it's really fundamentally it's the science that should be driving the effects of any type of nutritional supplements that you take, and we want everybody to be comfortable that the science is rock solid, and so we've really made that effort to create great clinical studies. As I mentioned before, our clinical studies are all double-blind, placebo-controlled studies, and and we've published them in all leading academic journals that are out there. So I think this is important for anybody working in this space to stay at this level of rigor.
Yeah, well, you guys have certainly been a model of integrity on that front. Let's talk maybe a little bit about delivery. I do actually have, first of all, I might also say that the packaging is exquisite. You guys did an absolutely beautiful job. So I have here, for those who are watching this on YouTube and not just listening to the audio, I have one of the packages here that I was lucky enough to receive. So this one is 500 milligrams of kind of like a powder, right? That just gets, you can, well, for me, I just put it in a glass of water, and it tastes good, but it's relatively innocuous. It's not a strong flavor per se. But I also know that there's a capsule version, I believe, and then I did actually try the shake version, which I actually quite enjoyed. It's sort of a protein concoction there with whey protein, I believe, but it was very good. I actually did it yesterday before my resistance training, and it was great.
Yeah, so we've, as you mentioned, Jeff, our thought was, it's important to take this in the morning, and most people have a ritual around their breakfast time, and so we wanted, when we started and we launched with the product that you just showed, which is this berry flavored powder, and we wanted people to have a food experience, basically, that they could control, and depending upon their preferences. If they, you can mix it into a yogurt. You can also mix this into your favorite smoothie. But the idea was, let's give people an opportunity to take a nutrition product as something other than a pill. And so that's why we introduced this product, and we've also introduced soft gels as well, and we have the protein shake, which has 20 grams of of whey protein that you were mentioning, which I take that. I took that just the other day too. It's great. Tastes great, and and in addition to that, we've recently taken a look at other delivery ways for urolithin A, and this is more not ingestible, but more topical. And so we've come up with, and this was a thought we had a few years ago of, what would be the impact when applying this directly on the skin. And because our skin is the largest organ of our body, as we all know, and it has cells, and it's it's influenced by our natural aging, our sort of intrinsic aging of our cells, as well as these extrinsic effects or just sunlight that we're exposed to that cause photoaging. And so we embarked on exploring, how can urolithin A potentially counter intrinsic and extrinsic aging. And so we've this last year we ran some studies on that as well, and what we saw is that we have an impact on also on both forms of aging when applied topically. So we see that following UV irradiation and application of Mitopure and our timeline formula, that we see a reduction in inflammation, and we've also seen basically a reduction in wrinkles as well when combined in our formula. And so we decided to introduce that just recently, and so that was launched just at the end of this last year, and we just started shipping that in the U.S.
Oh, wow. I wasn't aware of that. So is that in the form of a balm or a lotion, or how does one apply urolithin A topically?
So it's a cream, and so we have a cream that is we have a day cream, we have a night cream, and we have a serum, and so that's applied in the morning, it's applied at night, and so allowing you to get the same your cells to get the same benefits and energize your cells even from the outside.
Amazing. And it still has the same bioavailability or enough bioavailability to be impactful.
Yes. Well, what we've measured, we focused on measuring basically endpoints that are visible, like wrinkles, and which is what people are looking at when they look in the mirror, and also looking at inflammation following exposure to UV light. So we were glad to see that these benefits continuing when applied topically.
Yeah. So just back to the ingestible product for a second. So let's say I get on my routine and I'm taking 500 milligrams per day. How long does it take in terms of duration to actually see an effect?
In our latest clinical study, where we were looking in individuals over 65, what we saw was that after 2 months, we saw an improvement in muscle endurance in both the hand muscle, so this sort of first interosseous muscle, which is the muscle you use when you're opening jars and those types of things, and then also in the leg muscle. So we looked at a very targeted endurance of those muscles, leg muscle and the hand muscle, and after 2 months of taking the product, we see a significant improvement there. And then in terms of, I think I was mentioning before, the other study that were in a population of 40 to 65, these individuals saw improvement. We only measured after 4 months, and this we saw an improvement of greater than 10% muscle strength improvement after that time in the legs.
Got it. Nice. So this is not something that, you're just going to take one day and expect to see like an immediate change in subjective.
Yeah, it's not a stimulant, and I think that's
Right.
important. It's not like taking when we think of energy, you think of caffeine,
Right.
and which is sort of that instant stimulus, and uh, we like to think of this more like energy 2.0, and that you're fundamentally changing your cells and what's happening inside the cells, and those mitochondria are being optimized, and that takes time. Biology takes time. The cells need to improve, and then the cells, interact with one another, and then and all of that together takes time. And I think an easy way to think about, how long does it take to see an effect when you go into the gym and you exercise. How many gym sessions do you need before you start to see an improvement in terms of your muscle strength and your muscle performance? It's not just go to the gym one time. You have to do it on a consistent basis.
Right. Well, but we live in a culture that wants immediate results, right? So, but I think, any of us who have committed to a health journey or any journey, I mean, a learning journey, for example, you don't just build new neural networks, overnight. It's a process of continuing to stay committed to whether that's, learning how to play the piano or upgrading your mitochondrial function or hypertrophizing your biceps. So, but you guys have just done just such an exemplary job on all fronts, and you've really taken your time. You've done the research. You've packaged up, just as I said, a beautifully presented product, and now you're really helping upgrade a lot of people's lives, because as we touched on earlier in the conversation, what are we if we are not energy? Really at the core of what it's like to be alive is to create and burn energy. So you've gone right to the to the origin of the whole thing here.
Yeah. Well, thank you, Jeff. Yeah, we're really excited that, obviously when we started the company, it was hard to anticipate how things would evolve, and but with Urolithin A, we see this impact, and with all of, we get so much feedback from our customers, and impacts on different aspects of their life. I mean, it tends to be more energy and muscle focused, but it's different for each person, in that some people are mountain climbers, and they swear by it when they're, when they climb Mount Everest. We had somebody who climbed Mount Everest and sent us a picture of him on the top of Mount Everest with our package, and we said, "Is this real?" And the guy's wonderful, and has been very careful in monitoring all of the improvements that he experienced when taking the product. And then we have people who are cyclists, and people who are runners, and other, whether it's professional or just amateur athletes, or just normal non-athletic people. We've seen a lot of and heard about a lot of benefits, and so I think that's what's rewarding to us, is to being able to create something that people are feeling a benefit about. And I know everybody here at Timeline is really dedicated to that as a mission.
Yeah. So where can one avail oneself of the Mitopure Timeline product? Because as I understand it, is it only a direct-to-consumer offering, or where are you guys?
Yeah, we sell online on timelinenutrition.com, and if you go onto our website, you can have access to all of our nutrition products, as well as the topical products. We're also providing sort of nutrition to your skin as well. So
love it.
Yeah.
All right. Well, Chris, it's been a delight to get schooled in the mitochondria and the different things that we can do to upregulate our mitochondrial function, and specifically the Mitopure product and urolithin A. It's just fascinating. I think we're on the cusp of this new frontier where we can really have a lot of agency over our own health. I talked to my dad, and he's 80. Well, he'll be 81 next week, and his relationship with health was, for most of his life, was just going to see his primary care physician once a year, and he would take a battery test and be like, okay, yeah, you're fine, move along. That's it. And boy, things have really, really changed, and there's so many tools, there's so much data available now for people to really become, the CEO of their own healthcare, and you're playing a huge part in that. So, thanks for all the work that you're doing.
Thank you, Jeff, for having me, and it's great to it's great to be able to contribute in this way, and we hope that people can benefit from urolithin A, and that it will help them all reach their own health objectives as they work on them and try and optimize their own nutrition.
Nice. Well, to be continued. I'll see you in Topanga, I hope, sometime in the near future.
I hope so. Thanks so much, Jeff. Appreciate it.
Thanks, Chris.
Hey, thanks for watching. If you like this interview from the Commune podcast, then click subscribe and check out this video